Right now, human biology has shifted from an unchangeable blueprint into a field of actively editable software code. For years, Myriad Genetics held a monopoly on the BRCA1 and BRCA2 genes, charging patients roughly $3,000 for a single cancer screening. In 2013, the case of Association for Molecular Pathology v. Myriad Genetics reached the Supreme Court. The justices made a unanimous ruling. Naturally occurring human DNA is a product of nature. You cannot patent it. But the ruling contained a massive loophole. If a laboratory synthetically creates or modifies a sequence of DNA, that new sequence becomes a legally patentable human invention. Corporations didn't stop trying to own the genome. They simply stopped trying to discover life and started trying to engineer it. Because natural genes were suddenly off-limits, the biotechnology industry pivoted. The race was on to build and patent the ultimate genetic editing tools. In 2012, a collaborative team led by Emmanuelle Charpentier and Jennifer Doudna made a critical breakthrough by repurposing a bacterial immune system. Their mechanism, CRISPR-Cas9, uses the Cas9 enzyme as molecular scissors. It relies on two distinct guide RNAs to lock on to a DNA target. Then, they fused those RNA components into a single guide RNA, streamlining a bacterial defense into a programmable tool. They had built the ultimate biological scissors. But the billions in intellectual property didn't depend on the scissors themselves. It depended on figuring out exactly where they could be successfully used. The CVC team's initial breakthrough had a major limitation. They had only proven the mechanism worked in the simple, controlled environment of a test tube, using prokaryotic components, meaning simple, single-celled organisms without a defined nucleus. Meanwhile, at the Broad Institute, researcher Feng Zhang was running a different race. He wanted to apply these scissors directly to human biology. This diagram compares the predictable test tube environment against a complex human cell packed with chromatin obstacles. Guide argued human application was exceptionally difficult. Guide RNA had to navigate hostile pH levels and active RNA-degrading enzymes. Overcoming these exact barriers constituted a distinct invention. In the eyes of patent law, discovering a natural mechanism isn't enough. True ownership belongs to whoever successfully builds the tool for human application. To complicate matters, in March 2013, the United States transitioned its patent system from first to invent to first to file. Because the CRISPR patents straddled this exact timeline, the dispute triggered a grueling judicial dissection of vintage lab notebooks against digital submissions. Between 2017 and 2022, the Patent Trial and Appeal Board, the federal tribunal that decides patent disputes, repeatedly awarded the priority rights for human applications to the Broad Institute. But in 2025, the Federal Appeals Court abruptly remanded the case. They ruled that the Patent Board had applied the wrong legal standard regarding conception, the mental act of imagining and planning the invention. A decade of relentless legal gridlock makes one thing entirely clear. Human biological code is now treated as the world's most fiercely contested corporate asset. Because the Broad Institute holds over 30 key patents, any company trying to commercialize a human CRISPR therapy must navigate a massive bottleneck and pay heavy licensing fees. Those corporate legal costs flow directly to the patients. Modern gene therapies carry price tags that routinely reach millions of dollars per treatment. We have reached the era of the biological paywall. The scientific tools required to cure devastating diseases exist, but they are tightly restricted by proprietary monopolies. When life-saving genetic edits are locked behind corporate patents, biological survival transforms into a luxury reserved strictly for the highest bidder. Looking forward, human DNA functions as high-value training data, harvested to fuel proprietary AI models and corporate databases. We are approaching a fork in the road. Down one path is an open genome movement that shares biological knowledge as a public good. Down the other is a landscape of absolute corporate control. If your biological code can be rewritten, and a corporation legally owns the edits keeping you alive, who really owns you?